So welcome to World Myeloma Forum Expert Panel. It's a pleasure to have everybody here. We're going to try and review some of the abstracts from ASH 2017. So before we get started, just as a quick introduction for all of our viewers.
I'm Sagar Lonial from Emory.
Suzanne Lentzsch from Columbia University in New York.
Ravi Vij from Washington University in St. Louis.
I'm Ola Landgren from Sloan Kettering in New York.
Excellent. Welcome. So I think the first topic we want to discuss is much of the data around the CAR T cells, and there's 3 abstracts presented at ASH this year by Dr. Cohen, Dr. Kokendorfer, and Dr. Yan. So Sagar, your thoughts on the data around the CAR T cells and differences between the CAR T cells that were presented?
You know, it was interesting. I've been a bit of a skeptic on CAR T cells in myeloma, mostly because I've not been overwhelmed by the data in lymphoma and in ALL. And what I mean by that is it's not that I don't think they work. They clearly work. But the long-term PFS is not as good as what you get from the early response rates. And what I think we saw from certainly the Bluebird abstract that was presented yesterday was the idea that the response rate was even higher than what we've seen in almost every other subset disease for CAR T cells. And then at least with 9-month follow-up, the median PFS hasn't been reached, which is very different from lymphoma or ALL in terms of CAR T cell efficacy. So to me, that was really exciting.
I agree. I think the 3 abstracts provided evidence of proof of principle. So it works in certain patients. So we see responses, we see complete responses. There's a big question how durable those responses are. I think in multiple myeloma, we need to do a lot of groundwork. So those presented abstracts targeted different targets on the myeloma cells. So it's not quite clear whether BCMA or CD19 are the best targets. There was a question on BCMA, whether soluble levels of BCMA might interfere with those antibodies and neutralize the CAR T antibodies. There was also an abstract by Dr. Berenson suggesting that the BCMA level might be critical for antibodies targeting BCMA. So I think we need to do more fine-tuning on the right target, dosage, and also conditional therapy. But I think what we saw here was really proof of principle. It can work in multiple myeloma.
No, I would agree. I think that I echo that there is certainly clear-cut efficacy. What we lack is long-term follow-up to see how long this efficacy lasts. And I think the toxicity signal is somewhat different in the different presentations. The Bluebird does seem to be better tolerated, though they did talk about 1 case of CNS toxicity even with the Bluebird product. With the product from Penn, there was certainly more both neurological toxicity and CRS. And the data, as Sagar mentioned, with the Bluebird CAR does show sustained responses. But as far as the data from Penn goes, I think Adam said that it was median of 9 months as far as the progression-free survival goes. So that does, I think, bring a word of caution, because I see a lot of my colleagues here talking about trying to rapidly move these into even early lines of therapy, frontline therapy for high-risk disease. I think it is a good concept, but I think we need at least another year of follow-up before we make any concrete plans to go right up front with these.
I share those perspectives. I think I've already said all the key things. It's always, of course, hard to be the last person here to add something new. But maybe one thing in addition to what you have said, and I agree with, I think a big barrier is going to be how this could be delivered in the clinical setting. And I'm not only talking about the fact that you have to manufacture the cells. I'm more thinking about the actual practical delivery. So if you have to have patients in the ICU and you have to have them in the hospital, we are not really set up to do that. Yeah. So we are going to use resources in our institutions that we need for other purposes, unless we build any new hospitals everywhere. So that's not going to happen. So I think looking at some of the data when people are thinking of maybe spreading out and giving lower doses, maybe a couple of times, and try to reduce the CRS, I think that is also a very critical piece to think of how this actually could be implemented. So I agree, small numbers, short follow-up. If all holds true, I would love to see the data in a year at next ASH, but then the next barrier is going to be all those practical things, how it can be delivered.
No, that's a great point. So one of the questions is, with CAR T cells in large cell lymphoma and ALL, there was a little bit more of a clear-cut pathway because those patients, really beyond post-transplant, it's all chemotherapy, and there's not really novel therapies for those patients. And so the survival is short after second line, so there's a real space where the CAR T cells have really made a major impact. And it's different in myeloma where we have multiple novel therapies. These patients are living 5 to 10 years. So where would you fit it in because you have all these novel therapies? And so I think that's a little bit of a challenge where in large cell lymphoma, there's a little bit more of a clear-cut, post-transplant patients where you can potentially cure them. It's a little bit different in myeloma with so many novel therapies. How do you think it'll fit in because of that?
I wanted to point that out, with all our criticism CAR T cells, we should keep in mind that those were the sickest patients, So there is a waiting line to put patients on CAR T cell therapy, and very often patients are really relapsed multiple times. They are penta-refractory, they have no other options, so we put the very sick population on our CAR T cells, and that might be also the big difference between trials we use for kind of ALL, or for high-grade lymphoma. Those patients come much earlier into the CAR T cell therapy. You don't have an acute leukemia, 6 lines of treatments, and then you decide, after several months that you might kind of try CAR T cells. You have to do it early, in the first, second relapse. So that might kind of, be an issue that we have to bring the CAR T cells earlier. If it is safe and if we did more work on the fine-tuning, maybe kind of, we bring it earlier. If you talk to transplanters, sometimes they say maybe it substitutes transplant. I don't know. Ravi, you're a transplanter. What do you think?
I think that, again, the path forward in the short run I see is in the highly refractory population, but I share your concerns that that may not be the population that in the future may benefit most from the product. But I think that is where the product will probably first make FDA approval. And then moving into earlier lines of therapy, I think we can speculate on trial designs. I've heard people talk about trial design of Dara-Dex or Dara-Rev-Dex versus CAR T cells for more definitive phase 3 designs. Also people have talked about, as I mentioned, taking the high-risk group of myeloma, perhaps the stage 3 revised ISS, and doing a trial there. And again, that would be probably a single-arm study. I think. What do you think, Ola?
I think moving it up earlier is not very different from anything else we use. We have seen for years every drug that works in the relapse setting, if you move it up earlier, usually it's better there. So it's kind of the same thing. I think the key thing with CAR T cells is clearly the toxicity, and I think also the feasibility of delivering the drug. So I think all these things we have talked about, those are the barriers that need to be figured out. We need to see durability, we need to see how the safety can be improved, and then for the feasibility, that needs to be worked on. And then it can move up if all those things work out well.
Yeah, I mean, I think when you put it in the context of all the available therapies,
Yes.
the presumption is its long-term efficacy is going to be like anything else we give, meaning that the curves continue to come down and that eventually all patients will relapse. And I think the hope, if this is really going to make a big dent, is that patients will eventually be cured of their disease with this approach. And if that ends up being 25% of patients who get CAR T cells, that's a pretty big fraction, particularly in the relapse setting. And so I don't, if all it becomes is another salvage therapy for relapse, that the curve comes down like everything else does, then I don't know that it's a step forward. But if it does actually cure a subset of patients, then I think it's a big step forward, and we just have to understand who those people are.
I agree. I think that the bar has shifted. When the technology was first, advocated for ALL and non-Hodgkin's lymphoma, the hope was cure. Now, though the hope remains cure, I think we're even more accepting of PFS as endpoints in these trials. So we certainly, I think, changed our perspective in that regard. But I echo Sagar that cure is the thing that drives the field forward with this modality.
So just to kind of wrap up, I think interesting data, nice response rates very early on. We have to understand, are we going to cure patients? And that's really kind of the expectation or the bar that we have. If we can do it, can we actually logistically implement this? Is it going to impact many of our older patients, or is this going to be limited to a small subset, and how do we deliver that? So much to be learned, very exciting data in CAR T cells.